actually just needed 5 more minutes, but no, Holly's doing exactly what I would normally do. There you go, right there is perfect.
We need a plus over here.
Need some strike some marks.
Yeah.
The little piece of red carpet.
Okay, so it's my pleasure to introduce John Galeotti. He's going to give us a talk about some of the medical robotics and medical imaging work that he's been doing. He got his undergraduate degree from North Carolina State University, where he was valedictorian. Then he got a master's degree there a year later in 2002. He liked master's degrees so much, he came to Carnegie Mellon to get a second master's degree, which then was followed with a PhD in robotics from Carnegie Mellon. Since then, he has been working as a project scientist with various collaborators here in robotics and at the University of Pittsburgh. It's pretty impressive what he's done so far. He's been able to actually lead and obtain an NIH R01 award, which if any of you have tried doing that, you know how painful it is to just do it, let alone win. And right now he's in the process of securing another DoD project. So he has a lot of great things to talk about, and I figured instead of me talking about them, he can do a better job than I, so I'll sit down.
Thank you. So, as Howie said, I'm a senior project scientist here in robotics. I also have adjunct appointments with advising privileges in biomedical and bioengineering here at Carnegie Mellon University and at the University of Pittsburgh. And I direct the Biomedical Image Guidance Laboratory at both universities. So what do I mean by biomedical image guidance? Well, I mean research into the guidance of biomedical tools and actuators. And this covers everything from image acquisition and improvements to new developments in those technologies, image analysis, computer vision kinds of things, and also augmented reality visualization. The goals of this are certainly first and foremost in many ways to give better guidance to surgeons and better guidance to robotic manipulators. But going beyond that, I'd like to see and measure things that were not previously possible, working in the core imaging technologies themselves. And to go with that, to then be able to improve medicine and gain fundamentally new biomedical knowledge. So that's where I'm going with this. Doing image guidance does pose some unique requirements. Real-time operation is one of those. There's lots of things we do in computer vision and medical image analysis that work really well if you have a cluster of computers and 2 hours for them to operate. Lots of things that work reasonably well if you have 5 minutes on a desktop. Most of those don't work so well if you want them to run at 15, 30 frames a second on real-time data stream. There is a much tighter requirement for registration between multiple 3D coordinate frames. And unfortunately, patients are soft and squishy. Most things we want to do, especially if we're not talking about orthopedic surgery, involve soft tissues that are deformable. And so what registration means in that context itself is complicated. Deformable registration is usually not something we think of as real-time. And there are complicated issues here to deal with as we try to maintain the level of registration and alignment needed to safely guide tools to small targets with dangerous things sitting right beside them that we don't want to injure. Finally, in all of this, we have to develop devices that have geometries that are actually usable. We can't have our tools sitting right in the way of the patient where the surgeon can no longer get to where they need to get. And often this results in forcing us to do compromises, and we have to work to make those compromises acceptable and see what we need to see, guide what we need to guide, and still stay out of the way so that things can proceed. My core process in a lot of the work that I do is to begin with understanding the imaging modality at a sufficiently fundamental level that I can change it and modify it as appropriate to be able to make the system work as a whole. I then change, as I said, changing it to fit the application, and then I proceed from that to develop the entire system, an augmented reality system in many cases, or some kind of another perceptual and guidance system, but ultimately giving new forms of vision to humans and computers. And this work entails novel contributions to optics, computer vision, and psychophysics. So I'm going to, in this talk, begin by presenting 2 different systems that I'm working on. The first that I'll spend the majority of the time on is work for the R01 that Howie mentioned. And this is in progress, developing a microsurgical augmented reality guidance system. I will then talk next about the system that's just starting, And this is a system for which we're getting the DoD award. And this is for a multimodal approach to enhance clinical ultrasound with computer vision so that the ultrasound probe will know where it is located relative to the patient themselves without need for third-party tracking, able to hand deformation of the patient, and so on. I'll then discuss a little bit about my future research directions and finally have a couple of slides about the teaching that I do as well before wrapping up. Our microsurgical guidance work uses optical coherence tomography, OCT, to guide microsurgery. And this was funded by the National Institute of Health's Eye Center with the application of guiding ophthalmology surgery, and in particular on the anterior segment of the eye, which is the front portion of it. So cornea, lens, that kind of stuff. This is not for retinal surgery yet. That's future work yet to happen. In describing this, I'm going to talk some about the clinical needs and what we're trying to do here. I'll talk about the general development of the system, proceed to describing the novel optics work that was done in terms of the scanner and in terms of the visualization aspects, talk a little bit about real-time image analysis, and finally talk about psychophysics and understanding how humans perceive these images under a microscope, which it turns out is complicated. Our initial application for this work is treating glaucoma, which is the second leading cause of irreversible blindness worldwide. Glaucoma is believed to be, with strong but not conclusive evidence, caused by increased intraocular pressure. And we do know that reducing pressure in the eye does correlate with positive outcomes in glaucoma patients. One of the ways we can treat this is by treating the collector channels that drain intraocular fluids from the eye. And all of this happens in what we call the limbus, which is the region between your clear cornea and the white sclera of your eye. So you can imagine right around the ring of your iris, everything interesting all comes together. And you can see, right up here, you have a cross-section of the eye. The cornea comes down, and suddenly we switch to the white of your eye. And here's the lens floating inside it. And this little part right here is where everything interesting happens from our perspective in this regard. OCT can image this, and this little piece right here blown up is what you're seeing here with an actual real OCT scan. And this image was the first image in the world to conclusively visualize Schlimm's canal, this tiny dark region right here, which is basically a collapsed, flattened inner tube that circumscribes your iris. And it functions kind of like a ring network, if you will, but for fluids, that makes the interface between all the collector channels that gather the fluid from your eye, and that then lead into the channels that feed these fluids into your actual venous system so that you can have the exchange of nutrients and waste between your bloodstream and between the eye. Schlimm's canal can, in fact, be rotor-rooted, if you will, kind of like plumbing. And they basically take a tiny needle, they thread it in, stick Schlimm's canal, and thread the needle all the way around the eye. This forces it open, and this, generally speaking, does reduce ocular pressure and lead to improved outcomes. One of the problems with this is that you can't see Schlimm's canal. Even with the highest power surgical microscope you could use, it's not visible. These structures are generally one transparent structure inside of another transparent structure. There's no way to see them. OCT, on the other hand, as you can see, can pick up the different layer transitions here and see it. And so instead of surgeons poking about where it's supposed to be and trying again until suddenly the needle starts following this path without too much resistance and causing lots of damage in the meantime while they try to find it, they could directly aim for it, stick it, hit it, and have much less damage in this procedure. Other targets of interest for various procedures, the collector channels, scleral veins, and again, Schlimm's canal you can see right here. This image does not look very good on the projector, unfortunately. If I had a quality monitor in front of you with good contrast in the dark grays, you'd be able to see that more clearly. Other applications here, I do not have funding for this. This is going into future work. The palisades ovate are the epithelial stem cell niche in the cornea. These are really important. They produce the stem cells that let your cornea heal itself, regenerate. If you do corneal transplants, you really want to carry these with them. If you want to do science with ocular stem cells, these are important. But there's currently no real way to harvest these things, preserve them, do anything with them. The first person ever to see these things in OCT, Kira Lathrop, is one of my new collaborators in ophthalmology. And she and I plan to take the system we're developing and then proceed to tweak it to enable harvesting of these palisads so that we can actually enable new forms of clinical treatments, new forms of science and stem cells, and a wide variety of clinical and scientific applications here just by being able to see what we're doing with harvesting these, transplanting these, preserving these things. At this point, I'd like to talk a little bit more about what OCT is. Very briefly, I'm not going to go into too many details here. The basic idea of OCT is that we're going to shine light into a translucent thing, skin, tissue, whatever, and we want to see the structures inside. And to do that, we have to filter out all of the scattered light. So if you shine a flashlight into your finger, you see this vague red glow as the light all bounces around inside there. We don't want that. We want crystal clear images. The way OCT can do this is that OCT basically uses a very low coherence light source and a Michelin interferometer setup so that only the light waves that follow the direct path in and the direct path back end up constructively interfering and get preserved. All the other light that bounces around has a different path length, and so when it passes through the interferometer, we don't get constructive interference and we can filter that out. That's the very basic idea. It was invented by Fujimoto at MIT. My clinical collaborators were the first collaborators with him on this, co-authors on his first science publication introducing the world to OCT. And OCT can image 1 to 2 millimeters deep in translucent tissue. Beyond that, we have so little light falling or going in and coming back out directly that we basically have no signal, and signal-to-noise ratio gets horrible. There is an improved version of OCT that uses the frequency domain to be able to image all the different depths at once instead of having to adjust our reference mirror and our interferometer. And this is the kind of system that we're actually using. It consists of a diffraction grating, line scan camera, and so on, and is able to image an entire series of depths simultaneously using this trick. So our goal, as I said earlier, is basically to give X-ray vision to the surgeons. But in this case, to be able to see structures that aren't at all visible. In the clear cornea, for example, in OCT we can actually distinguish between the epithelium, Bowman's layer, the majority of the cornea, which is the stroma, and even see right down here this very thin line at the bottom, Descemet's membrane, and the endothelium. There are a lot of procedures for which we want to completely remove one of these layers and completely preserve the layer right beneath it. And again, in the clinic, they try this. Once in a while, they succeed if you don't have too much adhesion between the layers. But very often, what would hopefully be a simple procedure with low risk ends up having to be turned into a much more invasive procedure when, oops, by golly, we just punctured that membrane below. Now we have to remove it to transplant a lot of stuff we didn't want to, do a lot of patching. Things get complicated in a hurry frequently, and we don't want that to happen. With OCT, we could actually see what we're doing, and again, very carefully remove one layer while preserving the other. And then likewise, as I said, of course, access Schlemm's canal, access scleral veins, so on and so forth. In addition to the cornea and these things in the limbus, this could also presumably be used for various kinds of lens replacement surgery. That's not our focus because it tends to be more of a replacement procedure at the moment. There's not as many fine details they're doing, but there are potential applications there as well. Using OCT to guide surgery, we need to have some kind of augmented reality system. Previously, all OCT systems to date have basically been diagnostic systems. They put the OCT scanner right in front of the patient's eye. They acquire images taking as much time as they need. And then they show them magnified as much as possible on this big, beautiful 20-inch monitor, high resolution, where the surgeon can, or not the surgeon, the clinician can fuss over small details and individual pixel nuances. In our case, however, we want to project these images at life-size scale right inside the eye itself, perfectly registered and lined up, so that the magnification is happening not by putting this image on a computer monitor, but instead the magnification is happening from the microscope itself, the surgical microscope. So that the image appears in place, it's magnified exactly the same way that the patient's eyeball is, and very importantly, the optical distortions that the microscope introduces to the patient's eyeball are equally applied in exactly the same way to the virtual image. And if this doesn't happen, then we lose the registration between the two. Coordination breaks down, lots of nasty things happen. So we need the bad things that happen from the microscope to happen to our virtual image just the same way they happen to the patient's eye itself. Doing in situ imaging properly has a number of benefits. It results in much more accurate aiming. Because you have more accurate aiming, you're able to follow straight trajectories to targets, as opposed to most other systems where you aim close to right, but not exactly, and as you get close, your needle tip or tool tip gets close to the target, you realize, oh wait, I'm off a little bit, and you make a mid-course correction. Unfortunately, mid-course corrections, when your tool is partway through the patient's tissue, usually results in some undesirable tearing, stretching, nasty things that happen. And we want to avoid that. And finally, while it's certainly possible for people and clinicians to learn to anticipate the kinds of mistakes they'll make and work around them, when they do their learning and get better, as soon as you put them in a different situation where something's different, they basically have to relearn how to accommodate for the errors of the system that they're using. And when you have patients with diseases where this patient's a little bit different than everyone you saw before, this introduces problems. In fact, no two patients are the same to begin with, let alone in disease conditions. And in pediatrics, this is particularly problematic, where we have the wide range of developments, sizes, and so on. So prior systems that people have made for injecting images into a microscope basically relied on a head-up display approach. Very often, they would just inject a single flat image that would appear to be at infinity, always in focus, and do this for a microscope, often a non-stereoscopic microscope. And if you're interacting with basically a flat plane, and you know it's a flat plane, then you don't really need depth perception. There's only one plane you're operating at, and all you have to do is get a linear 2D transform to align these things, and you're good. Depth perception is kind of irrelevant. But when you're guiding complicated surgeries on three-dimensional structures, this won't work. People who've tried to work around this typically use a stereo vision approach. This is kind of like what happens when you go to the movie theater and see a 3D movie, or play a 3D video game and so on, where a system directly computes different viewpoints for your left eye, right eye. Your brain sees the differences in those images, uses stereopsis to infer depth from that, and produce a sense of depth accordingly. Now, these systems still have some major limitations in terms of a microscope. The most important of these, we believe, to be accommodation, where your eye focuses. As magnification increases, stereopsis tells you less and less about where something is located. The cue that becomes increasingly important with magnification is accommodation, because your eye is able to focus only at a very thin slice as magnification increases. And seeing whether or not two things are in focus at the same time becomes the dominant depth cue to know where they're located. Are they at the same distance? Are they different? Is my needle tip at the same location? How far away is it? Well, how blurred your needle is relative to the thing you're looking at ends up becoming the most important way to tell how far away it is. And if we use a stereoscopic approach, this doesn't work for us. The stereo image is always in focus, or never in focus, neither one of which is good. And so by putting the image correctly in place, the way that using our technique, all of these cues are going to be able to work correctly. We do this by using true optical virtual images. These are the kind of virtual image that's made by a mirror, that's made by a magnifying glass, or so on. An optical virtual image produces light rays that are indistinguishable from a real object had it existed where that virtual image is located. Your eyes focus on them correctly. Stereopsis happens naturally without any real-time computation. Your ocular convergence works. All of the depth cues work correctly, perfectly, and naturally with a virtual image, if only you're making a perfect virtual image. Now, it turns out that trying to produce these is for anything other than a one-to-one mapping with a mirror is a really hard thing to do accurately. You end up with blur, and that messes things up. But we can still optimize systems to make the residual error within an acceptable threshold, say a millimeter, microns, whatever kind of scale we're working at. And because we take one of these virtual images and inject it beneath the objective lens of the microscope, as I said earlier, all of the optical distortions of the microscope get applied equally to the virtual image as well as to the patient. And because of that, there's this perfect alignment that remains between the two. And we have the perfect focus or lack thereof between things that are at different depths. All of the depth cues work. The basic idea, represented simply here, is to have the OCT scanner imaging inside the patient's eye. And one way or the other, we use something like a half-silvered mirror to make a virtual image of a life-size scale image of the OCT scan such that the reflection appears down in the eye in the exact correct location. So when you look through the microscope, through the mirror, you see the virtual image floating in place. And this is the simplest possible way you could do this. We produced a couple of prototypes. This was the first. It actually did not produce a nearly bright enough image to be visible under the microscope. Microscopes expand the image and therefore dilute the light flux per millimeter. This is a reason why microscopes have bright lights on them. Unfortunately, bright lights don't help with a flat panel display by making it brighter because it has its own internal illumination. I led a senior design team to produce a new prototype which borrowed technology from rear projection televisions designed to have external light sources using a liquid crystalline silicon microdisplay. And using this approach, we were able to produce a very small, very bright, high-resolution image. In this case, you can see 3 very tiny metal pins here, the OCT image of the 3 of them. It has specularities happening at the cross-section, but you can see the 3 simple curves. And looking through the calibrated system, you can see the curves of the OCT are perfectly aligned with the 3 pins we're looking at, demonstrating that the system is calibrated, and they're able to see it correctly. I think the resolution of this display was 12 micron pixels, I believe, and could easily adjust the brightness to whatever magnification we needed. Proceeding with this, we were able to produce the world's first ever in situ image of OCT inside of an actual eyeball. In this inset down here, you can see an excised cow eye, and you can see the cornea here, the white sclera over here, and that limbus, the interface region, has a needle stuck through it. And this is a crude approximation, but to demonstrate for the imaging something like what's going to be happening in limbal surgery, trying to access Schlemm's canal, so on and so forth. This is the view through magnification. You can see the needles coming in. It's penetrating through the sclera right before we reach the limbus here. Here's the boundary right here between the sclera and the cornea. We can't see the needle here because it's under the opaque sclera. And then as soon as we get to the cornea, we can see through it, and suddenly we can see the needle again. This box right here is our in situ OCT image. If you look at that, you can see that these folds that were made in the surface of the cornea and sclera by inserting the needle are, of course, picked up by the OCT scan. The alignment is perfect. The OCT scan also sees the needle. These are all things that are visible. It's also picking up lots of little subsurface folds that are happening, distorting the tissue underneath the surface that you couldn't see with your naked eye. And with increasing magnification, if we oriented this differently, you could see internally into the limbal structures I was talking about earlier. This is the picture that landed us the $2+ million 4-year R01 grant that I'm currently working on.
So you're just projecting incoherent light onto the tissue, right?
Yes, the virtual image in this case is incoherent, yes.
OCT was to use coherent light so that it doesn't scatter and so it doesn't come back. Now you're projecting
different wavelength. Visible light is, OCT does not use visible light waves. It happens in the near infrared. Right. And so we can use the near infrared low coherence length OCT scanner simultaneously with dumping all of this light into their, in the visible spectrum. They don't interfere with each other. And furthermore,
saying interaction of light with the tissue that you're saying, illuminating it on.
and furthermore, the virtual image doesn't actually have light coming from in there anyway. The appearance of the tissue.
No, if you're sending some additional light onto the tissue,
yes,
that's going to get scattered by the tissue, right?
Okay, but we're not sending additional near-infrared light onto the tissue, and we're only measuring near-infrared light.
But you're projecting something on, so where is the light going? It's just onto a screen somewhere?
So the light that made the virtual image is coming from a screen that's external. And that screen is being viewed through some kind of a beam splitter, pass-over mirror, or so on. And so the light actually isn't even reaching the eyeball itself. We have 2 different beam paths here. If it was, it still wouldn't interfere, though. We have other stuff I'm not discussing now, trying to use structured light to do some other fancy things. And as long as the structured light's not in the near-infrared, we're fine. So we're working to develop a much better integrated system. Attached to the microscope, you have the microscope up here, its objective lens, a proper beam splitter with the projection system over here, which will ultimately contain real-time computer-controlled optics, dynamic diffractive optics, to enable tricks in terms of repositioning the virtual image. We have a custom OCT scanner attached with 2 different rotation degrees of freedom that both have a remote center of motion about the patient's eyeball itself, enabling us to image from different directions without getting in the way. Patient's eye in the virtual image. Currently, the system looks like this. We have the microscope here. The objective lens is right here. We have a box containing the beam splitter open on the bottom. We have an open aperture rotation stage here that is attached to an arm that holds our custom OCT scan head here. We then have our equipment here that is being used to generate our in situ virtual image. At the moment, it does not yet have the real-time dynamic optics in it, but it does have a key requirement for that, which is that we're using laser light, which is important when we're trying to do diffractive optics. And I'll talk a little bit later about some of the important contributions we've made to imaging with laser scanning projectors. And just to demonstrate, we can use that remote center of motion to swing the scan head out to one side, the other side. We can image all the way around the patient's eye to see any clock hour of it that we want to, while keeping the microscope stationary and knowing where the system is. To produce these virtual images that are very small and very bright, our approach is to repurpose a laser scanning projector. In particular, we're using Microvision's Pico-P engine. Some other people here use these things for structured light applications. The basic idea is they use a MEMS mirror to steer a 3-channel color laser beam to raster scan each pixel one by one in an image. And this is nice in most purposes because the image stays in focus from some short distance out to forever. In our case, we like it because it's using laser light with a relatively low, long coherence length. And it's very bright and very small. Now, in this figure, what we see is a contrast between a normal projector, like the one I'm using for PowerPoint right now, which has to be focused. And for a given projection distance, such as this one, the projector is only in focus at exactly that distance. If I move the projector or the screen forward or backwards, very quickly it blurs. And the pixel size going up here, or spot size, gets way too big. Your one pixel overlaps the next pixel. The spot size is larger than the pixel spacing. The laser projector follows this line here, where from half a meter out until basically infinity, the spot size is slightly smaller than the pixel spacing. And so each pixel doesn't overlap its neighbor. We have a nice sharp image. Now, unfortunately, Gaussian optics, that is optics using laser beams, have some unusual properties with them in terms of how the waste of the laser beams behave. And so while they put the waste here at half a meter to get this nice property going out, you can see that the laser beam suddenly gets larger as we go less than half a meter. And this is, we want to be operating right here, practically on this axis, at very small images, distances like centimeter or less. At which point, using our existing system, we'd have the entire, each pixel basically be superimposed on all of its neighbors and not at all usable. So we want to take this in-focus region here and somehow find a way to take a laser scanning projector and treat it like a regular projector where we can refocus it, but down in this region. We unfortunately can't do that using a single lens. And we can't do that because we have two different optical phenomena happening. One is the pattern of the laser beams themselves, how the image is spreading out, how the laser beams are pointed. The second is the beam width and profile of the individual laser beams. If we try to take the laser beams and refocus them down to produce a small image, we're never going to be able to get the laser beam also to have a small waist at that image location. The image waist is always going to be closer to the lens than where we're trying to image it. And the smaller the image we try to create, the closer the waist of the beam will be to the lens. And we're never going to get the waist small enough. Even if it's right by the lens, the waist will still be too fat to get high-resolution images. Our PhD student Sam Horvath has been working under my supervision to produce the world's first system for refocusing in LSP. And the key trick here is that we use two different lens groups to be able to gain degrees of freedom to independently control the orientation and path of the laser beams and the cross-section of those laser beams as well. And we arrange it in a special setup where the first lens collimates the laser beams, and the second lens then causes them to converge. And so the spacing between these lenses becomes meaningless in terms of the overall beam profile, or the image envelope, if you will, where the beams are oriented. But by changing this distance, we can control how large the beams are here, which affects the size and location of the beam focus point or waist over here. And with this separation, we're able to adjust the spacing between those lenses, our new degree of freedom, to be able to set the system up to be able to have a very small image produced with very tiny spots so that we now have non-overlapping laser beams. And looking at this in comparison to our previous image, where again, to remind you, we want to be beneath this diagonal line, which represents pixel size and pixel spacing matching. For any different lens spacing you can imagine, and we have a family of curves representing different lens spacings here, we can produce pixel sizes that are smaller than the pixel spacing for some range of sizes. At large images, we actually have some flexibility anywhere along here that we could produce an image of that size and have everything work. As we go to smaller and smaller images, we have a more narrow space to work with. But we still have a part of the curve that's going to drop below the focal line there, so that we can always produce these really tiny pixels, very bright, at a scale that's now measured in millimeters and going down to produce images that are half a centimeter or a centimeter on diagonal with HD resolution. Very, very bright. So now we've taken a laser scanning projector and we've defeated its normal purpose and made it so we can refocus it like a regular projector while still preserving the laser-based illumination properties, which are very difficult to achieve with other means. Most other means, trying to backlight an LCD with a laser and so on, introduce nasty diffraction effects, and we're able to bypass it using this mechanism. We've also developed, in collaboration with a corporate partner, a new novel custom OCT scanner designed for anterior segment surgery. This system has a few key properties. The first is that it gets around the depth limitation of most scanners. Because OCT can only scan through 1 to 2 millimeters of translucent tissue, most systems are designed to only image a depth or an image range in the depth dimension of 1 to 2 millimeters. However, the cornea is clear. We can easily image through a much deeper range of it. It's curved, so we're going to want to see a larger piece of it over a wider depth range. And beyond that, when we're doing surgery, we're poking on stuff and it's moving. And we don't want the entire thing we're operating on to jump out of the field of view just because we touched it. So we have many compelling reasons that we want a deeper imaging OCT scanner, and we don't have theoretical limitations against that because of the clear nature of the targets we're working on. Also, we want to have a nice long working distance here to keep the scanner away from the patient's eyeball to allow room for the tools we're using. Most clinical systems, they put the scanner right in front of the patient's eye. They get a high resolution image and they use it for clinical diagnosis. Is your retina thinning? Do you have something detaching? Is there some disease going on here? Our system's designed for guiding surgery. We don't need quite the resolution of the diagnostic systems. 6 millimeter resolution that we achieved in our system is more than adequate. That's the size of a red blood cell. And you can't see a red blood cell under a surgical microscope. And by relaxing a resolution requirement, we were able to produce a system that could stay in focus over this range of depths that we desire, that nobody else cared about. And we were able to produce a system that was in focus at a much longer working distance, which is hard. The closer the focal distance is to the lens of the scanner, the smaller the waist you can get on it. Pushing the waist out and trying to keep a waist that's focused over a long distance are both challenges, and they're hard to do. And we were able to produce some custom optics in here, carefully line them up with the galvos, pupils, and so on, to produce a system that could image with plenty sufficient accuracy for our purposes, as much accuracy as could be viewed. These images at a long working distance over a deep range to actually meet the needs of what we're trying to do. Additionally, unlike diagnostic systems, which have a focus on image quality, and it doesn't matter if the patient sits still for a half a second while you acquire a 2D cross-section image, in our case, we want to be doing 3D visualization, pulling out cross-sections. We want to be imaging volumes in real time. To do that, we used a unique arrangement with a custom repurposing of a high-end line scan camera and used some tricks with other special software and hardware to produce a system that at eye-safe illumination levels, but it's not going to harm the patient, can image at 128 kilohertz, A-scan rate, which translates to basically being able to do a 3D scan of 128 by 64 pixels in the transverse plane and 1,024 pixels in depth for an entire 3D volume. Now, this isn't like what you would be using with your typical laser rangefinder from SICK that is going to be giving you 2.5 dimensions, where it gives you a depth distance to the first target at every point. This is an OCT image. It's volumetric. We have data at every single point in space over that entire 6 millimeter range. Our pixels are basically 6 microns apart. And so we have a tremendous amount of data flooding in here, and a careful architecture to reduce the lag as much as possible to bring this data into a GPU, reconstruct the image, do image analysis with as minimal time or processing time and minimal lag as possible, and do the visualization suitable for injecting back into the microscope, all in real time, all on the GPU, so that we don't have a performance penalty in terms of moving stuff back and forth between GPU and CPU systems. You may be thinking that that's a really low resolution. And while it's certainly not as high as we might desire, it is sufficient for 15 volumes a second. And for most purposes, that resolution is perfectly acceptable, because most structures in the eye are layers. And in a layer, we care a lot about subtle differences in the depth dimension, where we have this 6 micron resolution. And because the layer is flat or gently curved, when you're at millimeter level scale, even the cornea is very gently curved. If we have a bit more coarseness in our samples, that's perfectly fine. We're sampling something that's curving like this, and if we get a sample, every bit of it, we can easily see and determine the curvature there. And what we really need is that separation between layers in the depth dimension, and we have that accuracy here.
So John, I have a question. Just question. 128 by 64 is a plane?
So if you took a cross-section orthogonal to the imaging direction, so cross-section this way, it would have 128 by 64 pixels in it.
And how many layers of those?
And we'd have 1,024 layers of those stacked on top of each other.
Okay, so it's 1,020 something times 128 times 64.
Yeah, so basically, that's, if you do the math for 128,000 columns in depth per second, that's what you come up with.
And you get that 15 times per second?
Yes.
Great.
So 15 fully populated volumes per second, coarse lateral resolution, very fine depth resolution. Okay. So we're working to implement automated analysis procedures to see Schlimm's canal, scleral veins, collector's channels, all these structures we were looking at. While you can see these in OCT, if you have it magnified on a monitor, most of these things, even under magnification under a microscope, are going to be too small to see still. And so things that we might not need computer vision to help a surgeon see that black tube there normally, now we need the system to automatically detect it using computer vision so that we can draw a big red circle around that centered on it, so that while the clinician can't see that because it's too small, they can see this bigger circle and know I just need to aim for the center of that if I want to hit this thing. So we have easy computer vision problems that are hard because we have a noisy image and because we're trying to do this really, really, really fast. Some of the first results that we've achieved here, this is on a phantom that has Scotch tape folded and embedded in gel. And if you look at this, you can see we basically have two bright lines here. This is the top and bottom surface of the piece of tape. And it was creased and folded, so we have a piece of tape here and then another piece of the tape here. Looking at this, you can see we have very low signal-to-noise ratio here. This isn't easy to see when we're doing this at very high frame rates. If we have more light on the camera, we can get better signal-to-noise, but as I said, our volume rate suffers. And using some unique pre-processing and special tricks, we're able in a very real-time fashion, real-time meaning each one of these columns processed again in 127 kilohertz, to be able to pull out most of the line segment boundaries of that. And then it's pretty trivial to stitch those together into a single closed curve. And you can repeat this process to find the next surface beneath it, go down, 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 and find these cross-sections of these surfaces. And you can look for the holes in these surfaces, highlight those, so on and so forth, depending on what we're trying to do. And that represents the work of BME at CMU master student Tejas, who is an incoming PhD student in the RI for this spring, or excuse me, for this fall. We're also working on psychophysics experiments to understand how these images are perceived inside of the microscope. As I said earlier, microscopes introduce lots of optical distortions to the reality. There's very little work that's been done on understanding these. In fact, in terms of meaningful work, there's basically one paper. And that paper was a PhD thesis from Cornell, which basically analyzed what's going on and said it's almost hopeless that things are distorted so much, people are really bad at perceiving where anything is under a microscope. And they primarily focused on analyzing what are known as the pictorial cues. And for example, if you look at the wall over there at a picture of a robot or a rover or so on, we have a sense of depth looking at that, even though the picture is completely flat. We don't have stereopsis, we don't have accommodation, but we can still, just looking at the picture, infer depth of things. The cues we use to do that are the pictorial cues. And under a scientific or non-stereoscopic microscope, that's basically all you have to work with. And they analyzed these, and they showed that in a great many ways, these things lead to massive distortions under a microscope. The way the world is scaling versus the pictorial cues leads to lots of differences. And one of the biggest of these is what's known as a gradient in various kinds of feature scales. So for example, if you look at that picture there, you see the dirt and you can see the small pebbles that are close to you. And as you look to the distance, all the pebbles get smaller and smaller and then blur away. And that texture gradient from large at close to small away is one of the most compelling pictorial cues. And that doesn't scale correctly under a microscope. And you can see that, for example, in this picture. Under a microscope, it wouldn't look that way. We, however, are working with stereoscopic microscopes that are designed for surgery, that allow accommodation cues. And so we want to study how those cues perform and how they interrelate with these distortions from the texture gradients. And so we developed the first of its kind in the world algorithm for producing image patches that have rich features in them, enabling your eyes to focus, accommodation to work, enabling your brain to find correspondences to do stereopsis. But at the same time, these are designed to have features across all possible scales you could perceive, from too large to see because it fills the whole image, to smaller than the resolution limit of your eyeball at that scale, so that you don't have any texture gradients, because no matter how you tilt this, change this, you always see a full range of possible textures there. In essence, we're creating something like a fractal, but instead of having this property of all scales at once along a boundary, we have all scales at once at all points in the image at the same time. What you see here is OpenGL renderings of two different image planes. One is flat, as one is tilted. And when you look at that, I presume that you can't really tell which is which. But in fact, the one on the left is flat, and this one is tilted 30 degrees with the top towards you. But nothing there jumps out about it. Now, and we've done extensive experimentation to show that nothing at all jumps out. There's virtually no information from these images you can use to tell which one's tilted and which one isn't. But if we turn on stereo shutter glasses to allow stereopsis to work, people become very accurate at knowing exactly which one is tilted and which way it is, and so on. Visual system suddenly works nicely. Stereopsis works perfectly. You can tell how it's tilted and do this to less than 15 degrees of tilt very accurately. We've then proceeded to take these images we've developed and validated, print them out at the highest possible resolution we can, and put them on computer-controlled rotating stages under a surgical microscope with controlled lighting to preserve exact lighting on the thing regardless of how we tilt it. And we're just now starting to study how people perceive these things. And while we don't have fully rigorous data that I can present on this yet, this is just starting, we believe it's reasonably safe to say that one of the key takeaways to start with is that people perceive tilt, position, and orientation of these images better than anything else we could put under the microscope. Because they lack the pictorial cues that are misleading, all that you have is a stereopsis, which works, and the accommodation, which works. And so you can actually tell how these things are tilted. You can pull out these subtleties. Now, these things still don't scale correctly because of magnification. Magnification doesn't even scale depth the same way that it scales the transverse image. But at least you can pick up subtle changes, and these changes are going to apply equally to your tools and to the image. Now, this has direct clinical relevance to our image overlays, because when we're putting a tiny patch of OCT image in the surgical field, that patch is going to be a tiny square with some OCT data inside it. It's hard to tell exactly how a tiny patch is tilted and oriented if you don't have more information around it. We had originally thought we could get around this by making a grid that you can turn off and on that gives you, this large image in the same plane as the OCT image and have a nice grid in it so you get all these rich features. Turns out that's bad. It actually confuses you. The fact that the grid doesn't scale the way it's supposed to messes you up and you misperceive the orientation. If we want to add any features to help perceive things, if this is the plane it lies in, we're putting some padding the image with other stuff, we want content like this. And furthermore, in the OCT images themselves, if we tweak the image contrast to highlight speckle, not highlight speckle, and so on, we need to be careful that we aren't accidentally introducing texture gradients that are going to be misleading. And so nobody's ever studied this under a microscope before. Nobody has analyzed how people perceive these things, and we're the first to ever be doing this. I'm studying human perception under magnification and how the different depth cues interrelate to one another. Now I'd like to move on to talk about the next system. This is the system for which we're getting our funding now, going into contracting shortly. And this is a system to give anatomical context to ultrasound data. Interpreting ultrasound data typically requires knowing where the probe is at all times. An ultrasound tech who's imaging you is looking at the computer screen and taking this probe and pushing it on you, twisting it, moving it around. And they know what's going on because they can look at you, see where the probe is, and they know exactly how they just moved that probe to get from this image to that image. On the other hand, if you've ever watched an ultrasound video, and by ultrasound video I don't mean fetal ultrasound where it's an easy image of a baby floating in space, but I mean something like liver biopsy, or liver imaging, thyroid imaging, normal clinical medical ultrasound outside of the pretty pictures of a baby domain. It's really hard to know what you're looking at. You see these blobby, noisy images with some tubes through them, and you see new blobby, noisy images with some tubes through them, and you don't know, did the probe move this way? Did the probe move that way? It's incredibly disorienting. And for that reason, the current clinical practice is to basically discard the entire ultrasound video, and all you keep are a few slices that the expert technician, when they're taking the image, said, this slice corresponds to this, believe me, I told you so. And that's all we record because it's impossible to make heads or tails of the rest of it. This obviously leads to shortcomings in terms of how we can go back and do post-processing, or follow-up analysis, legal ramifications if you want to analyze what happened. And it makes it a lot harder to do longitudinal studies because we can't really get the same image twice. Our approach is to use cameras to continuously monitor where the probe is relative to the patient's own skin and anatomical coordinates so that we can record where the probe was. We could play back videos that show the image as a 3D rendering floating inside a 3D rendering of the skin so that now you would see the ultrasound slices exactly in the locations as they moved through the patient. And suddenly, hey, I know where these images were taken from. I can understand them. Perhaps even more importantly, though, the computer can understand where the images are. We can do 3D volumetric reconstruction. And unlike other systems people have built that rely on cumbersome external optical tracking, we can actually tell when the skin was indented. The cameras can see that, hey, the skin right around the probe is suddenly indented 1 centimeter relative to the surrounding tissue. So now this image that I'm acquiring does not exist. It exists at this place in the world, but more importantly, it exists at this point relative to the patient's skin. And this is part of why there's been lots and lots of papers published over the past decade about tracking ultrasound to do 3D volumetric reconstruction. And virtually none of them have seen any kind of real meaningful clinical practice. Because while you can do a great job if you just barely touch the probe to the skin with gel and move it around, real ultrasound requires that you use the probe as an active manipulator to push and twist and get the probe in just the right position, get things out of the way to actually see what you need to see. It's very hard to get a good ultrasound image. And to get good ultrasound images, we suddenly can't be doing this nice surface-only thing. We're continually deforming the patient, and we need to take that into account if we have any hope of producing a quality 3D reconstruction of the data using quality ultrasound images. So our approach is to mount cameras directly on the ultrasound probe to give it this visualization so it can see where the probe is relative to the patient's skin. And this work is being carried out by former CMU BME student Ji-Hung Long, who's now a PhD student at the University of Pittsburgh. And doing this, we're able to track the probe and the trajectory of the probe to know how the skin was deformed, how we got there, and so on. Our initial main application of this, for which we're getting funding, is to study chronic rejection and nerve regeneration for hand transplant patients. DoD is interested in hand transplants, as you can imagine. Now, with any kind of transplant, chronic rejection is a major problem. And there's lots of ways we can try to monitor for it. The problem is all of the methods that would let you know in time to do something about it are not safe for patients that have compromised immune systems. And patients who've received transplants have immunosuppression going on, so their immune systems are compromised. We can do CT angiography, but we're dumping radiation into them, sticking needles into them, could introduce bacteria. We can do biopsies, which is even more likely to introduce bacteria and do nasty things. But if we did a biopsy and looked at the medium vessels and took that and looked at that under a microscope with staining, we can see various structures here that would indicate intimal wall thickening of the vessels, which is the first sign that chronic rejection is happening. Also, for these hand transplant patients, it's very important for the nerves from the host to reinnervate the transplanted hand as quickly as possible. Because until they reach the motor end plates, the nerves the muscles are continually atrophying, And if they don't get there in time, you'll have permanent loss. If you transplant just a small portion of the hand and life is good, then the patient eventually regains full dexterous motor control that they can pick up a bottle with one hand, twist the cap off with the same hand, and basically have full human control. That's one of the most complicated things you can do, as I understand it, in terms of fine motor control in a hand. It's something that monkeys can't do. It's unique to people. And they regain this full level of functionality. They regain full sense of touch, perception, and so on. It's basically a fully functional transplant. But because nobody can really monitor how these nerves are regenerating, it's really hard to do clinical studies in terms of which drugs can we give them to make the nerves regenerate faster. How can we change our surgical procedure to make this more likely? If we have to put a graft in place, what's the best way to do that? The science is an open-ended question that's starving for real-time feedback on how these nerves are regenerating. There have been recent advances in ultra-high-frequency ultrasound that can actually see the individual fascicles of a nerve bundle here. This is brand new. There are only 4 machines in the world that you can use to do this. And this is, again, only under IRB approval. They're experimental systems. And we have access to one of them. And our hope is that by looking at subtle changes in edema, swelling, so on and so forth, compressibility of these fascicles, that we can actually infer where the nerve fibers are and where they aren't along the length of that. This is going to be a giant study we're going to be doing. And likewise, the same scanner can image blood vessels inside the arm with such fine detail that it can see the intimal wall right here. And we can measure the thickness of this. Now, as you can see, the actual thickness varies naturally along this. And this is problematic because while we could take the scanner, this brand new system, and we could put it on you and get a measurement of intimal wall thickness, if we come back 3 months from now and get a new measurement, even a subtle shift in the angle of the probe will have us imaging a sufficiently different patch that we can't say whether any differences we measure are because, well, we just imaged a different place in the vessel and it was bigger, but so what? Maybe it was already bigger there. Or is this actually a meaningful change that's happened over time? Using our approach with the cameras, we will be able to tell where the probe is located, how the probe is tilted, how indented the skin was when that image was taken, and precisely get back to the same configuration so that we can actually tell meaningfully, the changes that we're seeing, do those correspond to something real, or is it just natural anatomical variation along the length of the vessel? And this would enable, for the first time, safe diagnostic measurements of chronic rejection. And we could use the system likewise for many procedures where we want 3D information on patients that we can't do otherwise for other things that are visible. For example, in an MRI scanner, these patients have metal shrapnel embedded in them. We can't do MRI. CT, again, has radiation, and these patients often have systems that have trouble regenerating, other clinical problems. Can't do MRI, don't want to do CT. And if we could enable 3D imaging with high-resolution ultrasound like this, this would be groundbreakingly huge for the treatment of these patients. Challenges with PROBESIGHT: the skin is deformable. We need reliable anatomical landmarks. And if we're working on patients that don't have lots of freckles, hairs, and other things, basically all we can rely on is skin pores and the folds in the skin. You think fingerprint, but on your arm. To be able to see those structures, we need large, high-resolution images. In essence, what we're wanting to do is visual slam 10 megapixel resolution, if you will, in real time. And hopefully you're chuckling inside at this point because we can't really do that. You're lucky if you're doing visual slam at VGA resolution. Our approach to get around this is to use a high-resolution a priori map. We have access to a special system in plastic surgery at UPMC that was designed for pre-planning on plastic surgery for face procedures and so on. And this system is able to produce 36 megapixel, high detail 3D color images with full shape and visual appearance of a face with sufficient detail that, as you can see, hopefully can see there, you can see the individual crease lines in the cheek there. They're all visible. The system basically uses 3 pairs of SLR cameras and stereopsis between all 3 of them, and then between the pairs additionally from different viewpoints to be able to run some advanced proprietary software of theirs that takes several minutes to take this instantaneously acquired image and produce the point cloud, overlay it, produce this image. You couldn't at all use this in real time. But we can get one fixed image and let it compute and give us this high-resolution 3D surface map. Once we have this, we no longer have to do simultaneous localization and mapping. We have the map, we just need to do simultaneous localization. A much easier problem. Still hard, but substantially easier. Furthermore, having the map gives us, for the first time, a meaningful way to talk about coordinates in a deformable way. As I said earlier, we need to do 3D registration, and Cartesian 6 degrees of freedom aren't sufficient to describe deformation. Well, the probe is there. Well, is the probe there relative, to their navel? Where is the probe there relative to the operating room? Where is it? Now we can say the probe is located at this exact point on our pre-acquired surface map with this exact orientation relative to it. And furthermore, this image was acquired with the surface map deformed thus. And now we can describe fully what it means for that probe location. We have a full 3D deformation field for the surface map plus position, orientation relative to it, and so on. So we can actually meaningfully describe everything we care about because we can take everything and relate it back to the surface map, and we can deform that surface map. And we've used that scanner to also scan a forearm. And we can create this surface model. You can view it from all different directions, see the details in it, so on. Using this, we still have to do a real-time visual search to map for what the image from the camera is currently seeing to where it is on that prior scan. If we know about where it is, and we hope to be able to do that for reasons I'll discuss shortly, we can then generate a whole bunch of renderings of that 3D surface model from the possible viewpoints of the camera. Well, the camera was about here relative to the patient, but maybe it was here, maybe here, maybe here, maybe here, so on and so forth. Generate a bank of these images, and we basically have a visual search about this search space for which one of these images best matches the actual image we're seeing from the camera right now. Once we know which image best matches, we know what simulated viewpoint was used for the camera to generate that image. And so now we know where the camera actually is relative to that 3D surface map. And we've thus localized it. And so we're looking for the best match out of there. It's a visual match image search that we're trying to do it very rapidly across a large number of images in real-time frame rates. Our experimental setup, yes?
I'm really sorry to do this to you. This is very interesting, but I should have warned you. We have like an hour of talk time and then a little bit of time for questions. I don't know where you are in your talk.
I'm getting close to the end of this and just future work. So if I could have like 5 minutes?
Yeah, it's perfect. Thank you.
Okay. So we have a setup with a dinosaur phantom. It has very rich skin texture on it, and it's static, nice for validation. We have optical tracking for validation, but we're not using it. And we validated that if we took a bank of images over all these different search spaces and use something simple like mutual information, that we do in fact get the best match at the exact correct location. The metrics fall off everywhere else. So if we get the best matching image, it will be the correct location of the camera. And we're working to get around the to address the real-time nature of this, initially by reducing our search space to ignore yaw of the probe, because probes have a rectangular cross-section, so it's hard to rotate them in one dimension. And initially to ignore deformation as well, because as computational resources improve, that'll naturally take care of itself if we can get it working for the others. We can do that at not real-time, but real-time we're ignoring that. And most importantly, we're attaching the inertial navigation system to the probe as well. And we do this to get an approximate answer for how the camera is moved to have a nice narrow search space. Now, the system, as I'm sure you're aware, inertial navigation systems suffer from drift. The error accumulates in a nasty way. However, because we only need to use the INS to get an estimate of where the camera is, and after that we do a visual search to find out exactly where the camera is, we don't integrate this error over any meaningful period of time. We have a very small amount of drift, but we can still do a tight visual search. We find exactly where we are and reinitialize our INS system to now be doing a visual search from scratch again, integrating from scratch again. And so we never let the error build up, using the best of both worlds. INS for its short-term accuracy and the visual match for its exact accuracy, but over a narrow search space. The INS system we're using was recommended to us by Ralph Hollis. He uses it on BallBot, I believe. And you can see that its rotational accuracy is much more stable attached to this phone than the built-in system of the phone. Of course, it costs more than the entire phone does, but it's much more accurate. And we've demonstrated that its accuracy is sufficient to see where the image is relative to the model as we rotate the camera around, without any other optical tracking going on here. So, future directions for my research. There's a lot I can do with OCT guidance. I can adapt it to retinal surgery. A key difficulty there is that we need to image the retina through the small aperture of the pupil of the patient. And if we had the ultrasound probe right in front of their eye, that would be have the OCT scanner right in front of their eye, that would be easy. But to pull it back for the working distance, that becomes hard. We basically have to share the beam path with the microscope, which itself is going to put a tiny lens right there to get around that so the surgeon can see through the small pupil. We have to integrate this much more tightly into the microscope. Several engineering issues there to address. We can also adapt the system for neurosurgery. In neurosurgery, there's a bloody field. It's very hard to see what you're working on. And neurosurgeons have communicated to us that it would be tremendously helpful if they could just know what tissue is there underneath this blood that I'm just about to cut into. And beyond that, if they could know how deep is it from this tissue to the next layer, that would be great as well. You know, if they're trying to cut through one little piece of gray matter and they don't want to cut it all in the white matter nerve tracts that are underneath it, if they could tell, hey, I've got 3 millimeters here versus 1 millimeter, that's tremendously helpful to them. There's a system I talked about earlier for the VOT epithelial stem cell niche, a whole new application there. Likewise, there's lots of potential for collaborating with manipulators people here, Howie, Cam, and so on, and trying to take this system and combine it with manipulation tools to enable finer perception and control of what you're doing. Also, I'm collaborating with a biophysicist at University of Pittsburgh to develop a new 3D magnetic trap. Basically, it uses microsphere manipulators controlled by magnetic fields. And my key contributions here are to be able to image where exactly that microsphere is in real time in 3D at 100 hertz rate. Now, to do that, there's two key things that I'm going to do that no one's ever done before with OCT. One is to do active beam steering so that we can image only the tiniest possible frame around the manipulator and move that in real time to track it. That's the only way to handle this. We have to search over a large range, and we can't do that in real time otherwise. And the second is we need micron level resolution smaller than what the light waves of OCT will let us see. And so we're going to do some unique oversampling, taking into account prior knowledge about what that microsphere is going to look like and how we're trying to control it to be able to determine sub-pixel resolution at spacing, even though we can't actually image it directly. So two key contributions I'm making here to enable this entire system. And the system would allow major new contributions in a wide variety of clinical applications, ranging from detecting cancer, preventing cancer from coming back, tissue biomechanical properties, tissue engineering, a large number of collaborators lined up that would want to use this system. Finally, PROBESIGHT, I have the DoD funding now, and we're going to use that not only for the purposes there, but to gather preliminary data that would enable us to write following proposals for compelling broad spectrum cases of nerve crush, sever injuries, everything from carpal tunnel syndrome to paraplegics. Monitoring blood vessels could now enable us to monitor systemic atherosclerosis for a wide variety of patients that have heart disease, trying to image the neck and figure out again, as the plaque is thickening, where exactly are those measurements taking, similar to the intimal thickness but at a much larger scale. And thyroid biopsy, where you have lots of tiny structures you need to avoid that are integral to and right beside the thyroid and others that we need to hit. And those are just 3 examples of a great many we could do. I also teach a course in medical robotics. It's cross-listed in 4 different departments across 2 universities. The course has grown from about 6 students when I first started teaching it about 6 years ago to 25 students each semester now, and I always have a waiting list of about 15 students trying to get in. But I can't handle many more because it's project-based, and I try to put substantial time into helping them all with their projects, which has been very useful for me. It's given me inroads into other faculties' labs and what they're doing, and put me in touch with clinicians and new work that they're doing as well. I've also received funding from the National Library of Medicine to further develop my course materials and to integrate ITK, the image analysis library I work with, which I've also had funding to help develop, with OpenCV, the traditional computer vision library. And this work of integrating traditional computer vision with traditional medical image analysis in a real-time context has laid an excellent foundation for my work with PROBESIGHT. I also get funding from ECE to help pay for teaching that. So, yes, said that. So in conclusion, I do my biomedical image guidance. This includes image acquisition. This includes the augmented reality visualization of it and the computer vision and image analysis that happens in the middle to make all of this a reality. I work with hardware, optics, software, and so on to build these systems. so that in the end, I'm making fundamental improvements to the vision of both humans and of machines as they try to understand, analyze, and see the biomedical world. Thank you. Are there any questions? Yes?
Yeah, so I thought your system of visual registration on patients was interesting, but as you pointed out, patients are squishy and they move and change. So one thing that you would have to account for is obviously the positioning of the arm or whatever you're looking at, but also over time patients will change.
Yes.
And oftentimes when they're on these drugs, they have a large change in body mass. So how can you account for stuff like that?
That's a very good question. And in fact, our application of hand transplants, when they initially do the surgery, you have tremendous swelling all around it, and that slowly heals over months. So yes, it is a hard problem. And the only way to properly account for that is to be able to take the vision of the exterior where the probe is and the ultrasound data itself, and we actually have to analyze both of them in our 3D reconstruction. So we're seeing the exterior and the interior. Doing the 3D reconstruction as the internal anatomy is changing is an open problem. There's another PhD student I'm working with I didn't talk about here who's working on some of the nuances of that, and we have some great ideas that I didn't want to put up in a lecture that's going to be published on YouTube. We have ideas. They're not fully proven. The degree to which they work remains to be seen. Once we get past about 3 to 6 months, I think there will be a lot less variability and we can still make major contributions if all else fails. But we do have a plan in place to try to handle the severe deformation case as well. And I hope a year from now to be able to stand up and give a seminar about how great and wonderful all of that was. Excellent question, though. Others? Yes?
So when you showed the picture of the tilted thing,
The image patch.
so you said it was stereopsis that was necessary to infer that it was really tilted. In other words, if we looked at it with our own eyes. However, that surprised me a little bit because we hear in school that 2 eyes, stereo vision. But the truth is, 1-eyed people walk around all the time.
Yes.
In 3D, which leads you to think it's rapid eye movements or something like that. That's giving you 3D view. So what's happening there? Why do you think?
So stereopsis is actually not the strongest depth cue. The strongest depth cue is when you see object occlusion. If I look at my hands here and I move over here and suddenly my backhand comes in and out of field of view, that always wins. If you see that happening, your brain will absolutely infer that this one object is in front of the other one because it occludes it. Right. If you have other depth cues at conflict, you can get tremendous headaches as your brain tries to make sense of what it's seeing if you deliberately distort reality. But that's the number one cue. Secondary cues, motion is always tremendously important. And then going from that, lesser things. The pictorial cues are important. Focal distance is important. In life, focal distance isn't usually as important. Under a microscope, it's much more important, and so on. So the key is that some of the cues that are really they're important but misleading under the microscope, we can try to suppress those so that the stereopsis, which is a weaker cue but much more accurate, can come forward and be used.
Right. That makes sense. Thank you.
You're welcome. Other questions?
Okay, if we have no more questions, let's thank our speaker.